So, Mr. Deepesh Joshi is from the Department of Molecular Biophysics Unit, Indian Institute of Science, Bangalore, and he is an integrated PhD student in the Molecular Biophysics Unit at the Indian Institute of Science, Bangalore, working in the area of vaccine development and immunogen design. His research focuses on epitope mapping, rational peptide, and protein design for SARS-CoV-2 and influenza viruses. With a strong emphasis on mRNA vaccine platforms, he has contributed to the design of variant-specific and conserved epitope mRNA immunogens, along with peptide engineering projects aimed at improving stability, safety, and immune recognition. He has secured top national ranks in major competitive examinations, including AIMS exam, where he got AIR1 in biochemistry, and the GATB exam AIR9, and your IIT JAM exam with an All India rank of 22, and your CSIR NET JRF and DBT JRF as well. He has also gained interdisciplinary research experience through summer internship at SCF Bio, IIT Delhi, where he worked on computational drug repurposing using virtual screening, molecular docking, and molecular dynamic simulations. Today, he will be speaking on the design principles and translational challenges of mRNA vaccines for respiratory viruses. So, welcome, Mr. Deepesh Joshi, and we are really delighted to have you with us. And over to
Thank
you.
you, sir. Thank you, sir. So, thanks a lot for the introduction. So, I hope my voice is audible.
Yes.
Okay. And so I will be sharing my screen and beginning up with the session. So, I hope my screen is visible.
Yes, it is.
Okay, great. So, great. Yeah. So, hello everyone, welcome to the session. And my name is Deepesh Joshi, as was introduced by sir. So, I am an integrated PhD scholar at IISc Bangalore, currently working in the Molecular Biophysics Unit. And my work is mainly focused on immunogen design. So, basically, I am working on immunogen design. So, that is to generate vaccines. So, basically, we use different approaches in our lab to generate potential immunogenic vaccines. One is protein subunit vaccines, another is mRNA vaccines. So, today we will be discussing about mRNA vaccines. So, as you would have heard about it at the time of COVID-19, that these mRNA vaccines they came into picture and they transformed the whole method of vaccination that we used to believe that you inject a protein that is immunogenic, that is any part of your virus, and then you can generate specific immune response. But generally now the approach is changed. It is more innovative and it is a better way of vaccinating people and to counter the future pandemics. So let's begin with the session today. So if we come over to the introduction part of mRNA vaccines. Basically, mRNA therapeutics, it is a promising alternative to conventional vaccine approaches due to rapid, inexpensive, and highly scalable manufacturing from in vitro transcription reactions. So, mainly what happens is that when you have to make a vaccine, the major challenge is its production and the scalability of the vaccine, that you should be able to have a mass production and you can deliver it to all the individuals who require this. So that requires a huge cost. It involves a huge cost. You have to purify the proteins. You have to, like, if you're making a protein vaccine, you have to purify the proteins. If you are making a inactivated viral vaccine, then you have to like do a lot of stuff to commercialize it. Okay. So, but in case of mRNA vaccines, mainly what happens is that you can simply carry out an in vitro transcription reaction and you can just begin with the mRNA production. Then another major advantage of this mRNA vaccine is that it is a non-integrating platform that is devoid of any potential risk of insertional mutagenesis. So basically what happens is, apart from mRNA, there is a huge you can say a new approach, that is DNA vaccines. So in case of DNA vaccines, what happens is it enters inside the nucleus and it gets integrated into the genome. Okay, so that is something that is an integrating platform, and it can have a risk of some kinds of mutations which can happen in the body of the patient when we administer the vaccine. So mRNA, it avoids those kinds of insertions and these kinds of mutations which can occur. So that's why it is a comparatively better platform than DNA vaccines also. It is highly translatable to produce an antigen of interest, and it is versatile enough for treatment of infectious or non-infectious diseases. Okay, so these are some of the advantages, and depending on the route of administration as well as the therapeutic goal, the efficacy of the mRNA vaccines are evaluated based on the expression ability of the coded protein. So before administering the mRNA into mice for studies or while we are researching on it, then first we do some in vitro assays. We check the expression of the protein that is coded by that mRNA in some cell lines, and then we get to know that whether or not that particular mRNA is worth carrying forward towards the in vivo studies. We can also look into the immunogenicity. Okay, so it is due to the recognition by the intracellular RNA sensors. Okay, and also about the stability and toxicity of the delivery system. So the design of mRNA vaccine majorly comprises of a mRNA sequence and a nucleotide modification. So as you are aware that a Nobel Prize has been given for increasing the stability of the mRNA, that is because of the incorporation of the modified nucleotides. Okay, so that is something which we will be discussing in this lecture. We will also be discussing about the optimization of the delivery system for efficient delivery of mRNA, because your mRNA, it is not something that can be directly taken up by the cell in huge quantities. It needs a carrier molecule or a delivery agent. Okay, some delivery agent which can deliver the mRNA into your cells, and then it can lead to efficient protein production. Also, the problem of the long-term storage and all we will be discussing. So coming over to a brief introduction, first we know that if we talk about the types of vaccines, we can have these inactivated vaccines, we can have these like protein subunit vaccines. So in case of inactivated vaccines, you majorly inactivate the proteins which are responsible for the causing the infection, or they are mainly responsible for the virulence of the virus. You inactivate those proteins and you inject that protein inside the body, and there are antibodies which are generated against that protein, and that gives you protection. You can also have some different kinds of live attenuated uh, viruses, okay, which you can use as a vaccine, in which you just attenuate the virus. You just put out some of the proteins, not the whole virus, to generate more immunogenicity. Also, if you talk about the um, so mainly there can be these different kind of viron-like particles which can act as vaccine, and among this you have this mRNA which can be used as a potential vaccine candidate. So how does this mRNA functions, and how does this mRNA is acting as a potential vaccine candidate? So the overall picture is that you have a mRNA and you encapsulate that mRNA inside these carrier molecules which you call as the lipid nanoparticles. Okay, so these lipid nanoparticles, they consist of a variety of chemical moieties, which can be your cholesterol, which can be your DSPC, dope, and other kinds of different lipids. Okay, and once the mRNA is encapsulated inside these lipids, basically this particle that is called as a lipid nanoparticle, this is injected into our body. The injection can be intramuscular or intravenous, but generally intramuscular is preferred. Okay, and once it enters into your body, these lipid nanoparticles, they fuse with your cells. Okay, and after fusion, so basically very important component of this LNPs is your ionizable lipid. Okay, so as soon as these lipid particles they enter into your cells, these ionizable lipids in the endosomes, due to a change in pH, the ionizable lipids they disintegrate. And once they disintegrate, they release the mRNA into the cytoplasm. Once the mRNA is released into the cytoplasm, this mRNA, this can now use the cell machinery to form a protein of interest, and it can be then degraded by the proteasome complex to make some peptides of around 7 to 11 amino acid long, and these peptides then they can be displayed, they can be carried by the vesicles onto the top of the cell, and they can be displayed on the cell membrane, and they can be then recognized by these T cell receptors. Okay, and once they are recognized by the T cell receptors, they can activate the cell mediated immunity. Okay, that is your T cell immunity. So the major advantage of mRNA vaccines is it can generate T cell immunity as well, whereas your conventional vaccines, which are protein vaccines or any other kind of vaccines, basically these vaccines they majority like you can say that primarily they generate humoral immune response only, that is the neutralizing antibodies. But mRNA vaccines, because of this processing and this peptide formation, it can be displayed on the cell surface, and basically with the help of this MHC class 2 receptors, it can bind to the CD4 positive T cells and to the T cell receptor of that, and it can activate these cells to activate the cell mediated immunity. So that is why that is the best advantage of this mRNA vaccines, as it can activate your cell mediated immunity as well as your humoral immunity. So here I have like differentiated this protein subunit vaccines with the mRNA vaccines. So majorly the vaccine component, that is your purified antigenic protein, and in case of the mRNA vaccines you are having the mRNA that is encoding the antigen. Okay, the antigen source, basically in case of protein subunit vaccines, you can produce it in vitro in the cell culture, in the yeast or in the insect cells. But in case of mRNA vaccines, it can be produced in vivo, that is by the host cells. Okay, so that is the major advantage, that the major antigen is being produced inside the cell using the host cell machinery only. Okay, the antigen delivery. So in case of this protein subunit vaccine, you can directly inject the protein and you can get some neutralizing antibody titers. But in case of this mRNA vaccines, you have to deliver it via a lipid nanoparticle, that is the LNPs. Now, if you talk about the antigen expression, so in case of this, in case of the protein subunit vaccines, there is no antigen expression in the host. But in case of mRNA vaccines, you have a host cell expression of the antigen, because the antigen is now displayed on the cell surface. Okay, and that is generating your T cell immunity. So both of these platforms, they do not enter into the nucleus, neither the protein nor the mRNA. But if we have DNA vaccines into comparison, then it enters the nucleus and it can cause insertional mutagenesis. So there is no risk of genome integration. If you talk about the antigen processing pathway, so mainly it is exogenous pathway, whereas in case of mRNA vaccines, it is endogenous as well as exogenous pathway. If you talk about the MHC class 1 presentation, then in case of mRNA vaccines, it is stronger. Similarly for class 2 also, it is stronger as compared to your protein subunit vaccines. If you talk about the CD4 positive T cell responses, then the responses are robust in case of the mRNA vaccines, and the CD4 positive T cell responses, they are stronger as compared to your protein subunit vaccines. Then if you talk about the antibody response, then it is similar in both the cases, but mRNA vaccines, they are more immunogenic because of the presence of like because of the activation of both T cell immunity as well as the humoral immunity. Okay, and mainly you require some adjuvant in case of your what do you call as these protein subunit vaccines to generate a specific immune response. But if you talk about your this mRNA vaccines, then usually adjuvant is not required, because the lipid nanoparticle that you are using as a carrier, that itself can act as adjuvant and can increase the immunogenicity of the whole vehicle that is being administered into the patient. If you talk about the innate immune activation, then it is minimal in case of protein subunit vaccine, but in case of mRNA vaccines, it is like intrinsic and basically mRNA plus LNP that is activating the innate immunity. If you talk about the antigen structure, so it may differ from the native conformation, because in case of protein subunit vaccines, we make a lot of mutations. Sometimes we delete the cytoplasmic tail to increase the expression, and we'll make a lot of changes in the ectodomain. We carry out some mutations to make the protein more immunogenic. So the conformation of the final protein, it can differ from the native protein conformation. But in case of mRNA vaccines, mostly the native ORF, that is the open reading frame of the protein, that is the same, and it is mostly native like conformation and processed by the host only. Okay. And the other thing is that when you purify a protein from a different host, because of the variation in the glycosylation patterns as well as some different variations in the post-translational modifications, the conformation of the purified protein can differ from the native protein conformation. Now, if we talk about the duration of the antigen presence, so it is short in case of the protein vaccines. In case of mRNA vaccines, it is transient. It is like the mRNA, they remain in the body for a very lesser period of time because these LNPs, these have a size of around 100 to 120 nanometers and they're easily like cleared off by the kidneys. But so the like time of the antigen exposure is transient, but it is sufficient to generate us like sufficient amount of immune response. If you talk about the booster requirement, so basically for protein subunit vaccines, you require multiple boosters, okay, like dose 1, dose 2, and all these boosters you have to take on specific intervals of time. But in case of mRNA vaccines, you require fewer boosters, okay, because the immunogenicity generated after the prime and the boost, they are like sufficient to protect you against the virus. If we talk about the safety profile, it is good for both. And if you talk about the manufacturing speed, then like mRNA vaccines can be manufactured much fastly as compared to your protein subunit vaccines. Cold chain requirement for protein subunit vaccines is low to moderate because in case of protein subunit vaccines, you can generate thermostable proteins which are stable at a higher temperature, also like 37 degrees Celsius. But in case of mRNA vaccines, you require a proper cold storage or a cold chain because like the mRNA, as we all know that mRNA is a very you can say a kind of very notorious molecule because it is prone to degradation and you need to have proper temperature as well as other requirements to preserve it inside the LNPs also. If you take examples of the vaccines, then you have the HPV Novavax for COVID-19, Novavax for COVID-19. In case of protein subunit vaccines, in case of mRNA vaccines, you have the Pfizer, Moderna, and all these kinds of vaccines. Now, when we were talking about the lipid nanoparticles, so now the question arises that all these lipid nanoparticles, what are they made up of? Okay, what is leading to uh, giving them the ability of acting as a carrier molecule to carry the mRNA? So we will understand the chemistry part of it. We will understand the delivery part of it. Okay. So now first let's look into the components that are required to form the LNPs. Okay. So majority like majorly you are having these four components. One is your ionizable lipid. That is the most important component that is present. The ionizable lipid in case of Pfizer mRNA vaccine it was ALC0315 and in case of Moderna it was SM102. So the mm, what we call as the what is the role of this ionizable lipid is that during the mRNA LNP formulation you use citrate buffer and that citrate buffer it consists of something that you call as a lower pH, that is the pH around like 2 to 3, okay, and at that lower pH what happens is that these ionizable lipids these are positively charged and the mRNA is inherently negatively charged due to the negatively charged phospholipid backbone, and as a result of it, it can lead to encapsulation of the mRNA inside the lipid nanoparticles, and as soon as they are encapsulated, what happens is that it facilitates the endosomal escape when it is entering inside the cells. So that is a major advantage of the ionizable lipids, that because of the variation in the pH, it can encapsulate mRNA, it can disintegrate, and it can release the contents present inside it when it is required. So that is why ionizable lipids are the most important component. Then you have the cholesterol. So as we know that cholesterol is also present in our cell membranes, it modulates the membrane fluidity as well as integrity. So the proper ratio of the cholesterol that you have, that is very much required, because if you are having too much cholesterol then the membrane can be like too much fluidic, and if the membrane is like fluidic, then it can easily release the content anywhere, anytime. Okay. And if the cholesterol levels are lesser, then the membrane will be rigid, and the contents of the membrane, like the contents inside the membrane, that will not be released as per the given condition. So the levels of cholesterol play a very important role. Then you have this phospholipids, which improve the LNP stability and enhances the fusogenicity. So in the phospholipids, you have this DOPE as well as this DSPC. Okay. And then you have the pegylated lipids. So PEG, that is there, it is a hydrophilic molecule, and as per the FDA guidelines, PEG has been used in a lot of antibody drug conjugates as well as a lot of drugs. So PEG, it increases the systemic circulation and prevents the particle aggregation. Let's say you are having two LNPs, there is a high probability that these LNPs can aggregate, and once they aggregate, they can just um, destroy your entire formulation. So to maintain or to prevent the particle aggregation as well as to increase the systemic circulation of the LNPs, because if the LNPs, they are coated with the PEG layer, that is polyethylene glycol, and it is hydrophilic, then it will increase the systemic circulation because it will prevent the interaction of the LNP proteins. We can say interaction of the LNP with the blood proteins, okay, which are present like albumin and all those proteins. So PEG increases the systemic circulation. So that is why it is very important. And all these four components, they make up the LNPs which are present. Now, there are some challenges to generate adaptive immune response by mRNA. So one thing is that mRNA, due to its dynamic secondary structures or the double stranded part, because you know that mRNA consists of some complementary regions, or it can consist of some GC rich regions, because of which it can form a double stranded structure. Okay. And once it forms a double stranded structure, it can lead to what you can say as the activation of the toll like receptors, that is the TLR3, that is present on the endosomes. Okay. And when the TLR3, they are activated, they secrete, or these activation markers like INF1 or TNF alpha, and when these are being secreted, then it inhibits the translation of the coded antigen. Okay, so that is something which leads to a decrease in the adaptive immune response of the mRNA. So this is a major challenge to generate what do you call as a to generate adaptive immune response and to enhance it, because due to the sequence of the mRNA itself, it can be a major issue that you are getting a lesser immune response. So now, like, there are a lot of progress in this field to optimize the open reading frame of the mRNA and to generate mRNAs which are better translating, which generate a huge immune response and which can give you a better amount of protection as compared to any other vaccine. So there was a paper which tells about some kind of modifications of mRNA. So as we know that uh, with the recent Nobel Prize, it has been said that if you incorporate N1 methyl pseudouridine, okay, so you know about uridine that is naturally present in the mRNA, okay, but if you incorporate this N1 methyl pseudouridine, then you can increase the expression of the mRNA and you can prevent the activation of the innate immune responses, and you can increase the immunogenicity of your mRNA. So this has been like said that if you are using a modified mRNA, okay, then you can easily be able to generate a better immune response, as well as you are able to get a better response. So, but apart from the N1-methyl pseudouridine modification, there have been other modifications like the 5-methyl cytosine. When you are attaching a methyl group at the fifth position of the base, then you can also like generate a better mRNA that can lead to a better immune response. Okay, so that is a thing. And so in this paper, they have told that the sequence engineered mRNA without chemical nucleoside modifications, it can also enable an effective protein therapy in large animals. So this paper is breaking a previous notion that you need modifications in your mRNA sequence to get a better, you can say, expression of the protein. But in this paper, they have disapproved that. They have said that if you are having a sequence engineered mRNA, that is the ORF, open reading frame of the mRNA, it is designed like in such a way that without any modifications also it can act as an improved therapeutic agent. So here you can see that here we are having different doses of the mRNA LNPs which are being lipoeffected in the HeLa cells, and we are having different doses of 550 and 500 nanograms, and we are having three different kinds of mRNA, that is the unmodified, that is the one with the N1 methyl pseudouridine modification, and the one with both modifications, that is the N1 methyl pseudouridine as well as the 5 methyl cytosine. So mainly here what you can observe is that as soon as these nucleoside modifications, they reduce the protein expression in the sequence engineered mRNA. That means that in case of these modifications, the expression is lesser as compared to the unmodified mRNA sequence. And this like negative effect of nucleoside modification reduces mRNA with lesser effective regulatory sequence interference between the chemical modifications and the regulatory RNA elements. So that means that these modifications, they weaken the RNA protein interaction, and the pseudouridinate impairs with the IRES sequence. So that is something that they have claimed in this paper. And also they have said that if you are having this chemically modified mRNA, it has stronger limitation of maximum translation ability due to inactivity of the regulatory sequences in presence of the modifications. So mainly you can see that if you're having the unmodified RNA, that is the basic mRNA without any sequence optimization. So if you are having no sequence optimization, you are having a regular sequence, and then you are comparing the regular sequence with the modified N1 methyl pseudouridine made mRNA, then you are having a better expression. That is, they have said. But if you're having an optimized sequence of mRNA and then you are modifying it also, then you are having a lesser expression. So that is something which you can conclude from this entire paper. And it breaks our notion that the you should also have an optimized sequence. That is also important as rather than only the modifications of the bases. Now, key mRNA structure elements, we all are aware of it. So you are having a 5' cap, you are having a 5' UTR, then you have this open reading frame, that is the gene of your interest, which will code for the particular protein that is specific to any particular viral protein. And you are having this 3' UTR as well as the poly A tail. So the 5' cap, so it increased the capping efficiency, the cap structure, innate sensing, as well as protein synthesis. Okay, and overall it leads to increase in the stability of the mRNA. So you can optimize the various sequence of these elements. For example, if you optimize, so basically this poly A tail also, it is responsible for the stability, and the majority of the optimization, it happens in the ORF, where you can do codon optimization or sequence modification and increase the protein expression. Okay, so basically this is the open reading frame in which you can incorporate certain mutations. And now there is a lot of development in the field of AI/ML, in which you are having a lot of graphical neural networks as well as your um, CNNs and all different kinds of neural networks, in which you just put the sequence of the mRNA, and based on a database of highly expressed genes, as well as based on the database of highly expressed what you can say proteins which are coded by the mRNA, it can optimize the sequence. Okay, and for example of some of the databases is RiboNN, you can check it. Okay, and you can read about it, so you will get to know that it helps in generating optimized our mRNA sequence, which we can use for like making antigens, highly efficient enzymes, peptides, antibodies, bispecific antibodies, as well as nucleases. So the key game is to optimize the sequence of the open reading frame as well as the UTR. So this has been seen in these kind of AI models that the majority of the role in the protein expression is to do with the UTRs because they are the regulatory elements. So the UTR sequence needs to be optimized to a greater level, and then comes your open reading frame, that is the gene of interest, and it can be modified based on some mutations, or you can say based on some stabilizing mutations, so that the protein that is coded by this open reading frame, it generates a better immune response. Now, we need a suitable delivery system for our mRNA because, in spite of very promising therapeutic agent, mRNA has to overcome certain extracellular and intracellular barriers following the systemic administration. So, what are the challenges which are faced by the naked mRNA? The first challenge is that when you inject the mRNA, so what happens is that you deliver the mRNA. So, once you deliver it, there can be some extracellular barriers which can these are the macrophages, the albumins, and the serum endonucleases, which can lead to degradation of the mRNA LNP, and they can lead to rapid clearance of it, and they can interact with the LNP and lead to its disruption, releasing the mRNA into the bloodstream, and that can have various other effects also. Another thing is that there are certain intracellular barriers because once the mRNA is endocytosed and it is released inside, so there are certain barriers like what if there is no proper endosomal escape? What if the endosome doesn't disrupt? Then your mRNA will never be released and it will not give any particular immune response that is required. So that is something that we have to keep it in mind. And then coming over to different vectors, so they have been employed to protect mRNA during the delivery, which are viral vectors, which possess an innate machinery ability to overcome cellular barriers, but have difficulties in their construction and the scale of production. And these viral vectors, there is a risk of generating immune response against these vectors only. So that is something that we have to look into. Okay. And you also have these non-viral vectors. They are like the superior for mRNA delivery because they will not have their own immune response and they will be better in delivering the mRNA and by like to overcome these barriers which are there. Okay. Now, the major components. So I told you, you have the ionizable lipid, then you have the cholesterol, and then you're having the phospholipids as well as the peg anchored lipids. So if you see the cationic ionizable lipids, it constitutes 30 to 50% of the whole LNP. The sterols, they constitute around 20 to 30%, and the phospholipids, they constitute 10 to 20%, and the peg lipids, they are around 5%. If you see in the cationic ionizable lipids, you are having the SM102 as well as the ALC0315 in the presence of the acidic pH of the citrate buffer in which the mRNA and LNPs are formulated. They generate a positive charge because of which they are able to encapsulate the negatively charged mRNA. Okay, and then you are having in the sterols, you are having this cholesterol as well as the beta cytosterol, and in case of phospholipids you have this DSPC and DOPE, which are required to maintain the integrity of the LNP, and for higher systemic circulation time you are having this PEG, okay, which you can use. And then so now there are different lipid based vectors which can be used in case of mRNA LNP vaccines. So you can have these liposomes, you can have these lipoplexes, okay, and you can also have what you call as our traditional lipid nanoparticles which are there. So what are these liposomes? So these liposomes, they are these spherical particles which are made of one or more phospholipid bilayers, okay, and their hydrophilic head, it faces outwards. Okay, the hydrophilic head, it faces outwards as you can see in the schematic diagram, and the hydrophobic tails, they faces each other. So they enclosed a water filled aqueous core, and in this core you are having your mRNA which is present in the aqueous solution. So mRNA is dissolved in the internal aqueous compartment, and what you can see as mRNA is hydrophilic and negatively charged, so it stays dissolved in the aqueous core and it doesn't interact strongly with the lipid membrane. The limitations of using liposome is that basically it has a low loading capacity because the aqueous volume is too high. Then there can be chances of leakage, that is the bilayers are permeable over time. There can be some oxidative instability, that is unsaturated lipids, they can oxidize easily, and there can be a poor endosomal escape because of phospholipid bilayers, they are like too stable in this conformation. So liposomes, like they are excellent for smaller drugs but inefficient for large charged molecules like mRNA. If you talk about lipoplexes, Basically, these lipoplexes, they are electrostatic complexes between positively charged cationic lipids as well as the negatively charged mRNA. So if you talk about the structure of these, the mRNA binds to the surface of the lipid assembly and it forms a multi-lamellar sheet-like aggregate. Okay. So if you talk about the chemical reasoning, so if you think about it, so there is a strong forces of attraction between the cationic lipids as well as the mRNA. And there is no true encapsulation because mRNA is present on the surface of these particles and it is surface exposed. So there is a high chance of the degradation of this mRNA as well as there is a strong chance of the interaction of the mRNA with the serum proteins. So that is why also, there is a chance of aggregation and toxicity because these cationic lipids they disturb the membrane and it can lead to toxicity. And there can be also chances for inflammation and cell damage. So these lipoplexes they can load mRNA, but it is too aggressive for your body to bear them because they can cause a lot of complications when they are administered inside your body. So these are some of the disadvantages that I have written over here. You can read them. And then comes the lipid nanoparticle. So the lipid nanoparticle, they are not hollow vesicles as like liposomes. They are mostly devoid of any internal aqueous environment. They are dense, electron rich, and like the core is electron rich and the mRNA is trapped inside the inverted micelles. So basically you can see these micelles inside which your mRNA is interacting with the head groups and it is trapped. So ionizable lipids, they become positively charged at low pH and mRNA is wrapped inside the lipid micelles and it forms a compact protected core. So they have a high loading efficiency. Basically, they are charged neutrally in the blood because the pH of your blood is around 7.4, so it is neutral there. And there is a efficient endosomal escape because in case of acidic endosomes, it destabilizes the membrane and it can lead to rupture of this LNP and then release the contents of the mRNA. Okay. And since there is a no permanent positive charge on the LNP surface, it is less toxic to the body. So that is why LNPs, they are the best means to deliver your mRNA. That is something which you can conclude. Now, if we talk about these different groups, so you can see the structure of these lipids, the ionizable lipids, if we talk about the rational design of the ionizable lipids. So mainly these ionizable lipids, basically you are having the head group which controls the charge and the RNA interaction. Okay, you are having a linker which is present and represented here in pink color. So this controls the stability and the biodegradability of the mRNA. You are having a hydrophobic tail which can be saturated or unsaturated. Okay, and these hydrophobic tail they can control the membrane interaction as well as the fusogenicity. So if you talk about the head group, what are the key factors you have to look into while you are designing the head group? So basically, there are different nitrogen containing head groups. They can be primary, secondary, tertiary, or quaternary amines. They can be guanidine, pyrimidine, and imidazolium. So these are all the different kinds of groups which can be there, which are positively charged, and they can be used as the head group. So what is the chemical role of the head group? Basically, the head group, it determines the pK value of the overall lipid. It determines the charge density because of the positive charge in it. It determines the hydration as well as it determines the hydrogen bonding with the RNA. Now, so now you can ask me that, sir, why the charge matters? So the thing is that mRNA, it is polyanionic and the electrostatic attraction is required for the encapsulation. That is why it is important to have a positive charge. And mainly if we talk about the formulation, when we do when we have a pH of around 4 in the citrate buffer, then but the formulation means that we are mixing the mRNA and the lipid with a proper mixture in particular pressure conditions. When they both combine, they the mRNA that is encapsulated inside the lipids and it forms an LNP. So that is something we refer to as formulation. And in this case, the lipids they are dissolved in ethanol, that is organic solvent, and your mRNA is present in citrate buffer, that is your aqueous solvent. And when you mix both of them, then because of the citrate buffer, it has a pH of around 4. Then what happens is that these on head groups, they are strongly protonated. But in case of blood, when you administer the LNP, then their pH is 7.4. They are like neutral, and the overall charge of the LNP is neutral. Okay. But in case of endosome, when it enters inside the cell, the pH is around 5 to 6. Then it is again protonated and it can lead to disruption. Okay. So this is something that is important. And if we talk about the what you call as the terminal hydroxyl groups which are present. Okay, so you can see that you can have some terminal hydroxyl group which can reduce the excessive hydration and enable hydrogen bonding with the RNA phosphates. Okay, if you now talk about the linker design, so I told you that the linker is responsible for the stability or the biodegradability of the overall LNP. So what do you mean by biodegradable linkers? So if you have read a bit of chemistry, you would know that these ester linkages which are present, okay, so they are easily breakable and you can easily degrade them and you can easily clear them off. Okay, so that is why ester linkages are preferred in the linker. And mostly you can have different other kinds of linker as well. That can be ether, that can be carbamate, that can be esters, disulfides, amides, ketal, and phosphate or urea. Okay. Now we talk about the tail part. So the tail, if you talk about mainly, it impacts the pK of the overall LNP. Okay, so they determine the lipophilicity, the membrane fluidity, and the fusogenicity, the packing density of the LNP, and also the endosomal disruption. So you can have two different kinds of tails, as you can see in the schematic. You can have saturated tails as well as you can have unsaturated tails. So if we talk about the saturated and unsaturated tails, so the saturated tails, they can pack tightly. They are rigid membranes. There is a poor fusion. Okay, if you use the saturated tails, but if you use the unsaturated tail, you can introduce some kinks. That is, you can these kinks, they can be helpful in disrupting the endosomal membrane and releasing the contents inside the LNP into the cytoplasm, or it can like promote membrane fusion as well as create packing defects. Okay, so you can use the mixed tail length. Somewhere you can use saturated, somewhere you can use unsaturated. Okay, so if you are using like two tails of unequal length, you can create voids in the lipid packing and you can reduce the rigidity and increase the intermembrane mixing. So this enhances the endosomal membrane disruption as well as the release of the mRNA. So this is something that is important. And there are some in silico design parameters for designing the ionizable lipids. That is something you need to know. So one is your CPK, that represents or determines the ionization window. Okay, and you are having this C log P that reflects the lipid's ability to partition into the membranes. So majority, like you can have a conclusion that the CPK value should range from 9 to 10.5, the C log P value should be around 15 to 20, and the C log D value should be around 10 to 14. So these are some of the parameters like you can look into while designing your tail part of the LNP. Okay, now like there is a role of other lipids that I have told you. So you can see over here that pegylated lipids, they provide LNP with an exterior polymeric layer, okay, to hinder the adsorption of the serum proteins on the LNPs and the mononuclear phagocytic system, extending the circulation time in vivo and prevents aggregation of the nanoparticles in storage as well as in the bloodstream. So the amount of pegylated lipids may determine the particle size, and the particle size is something which is of huge concern for us, because if your LNPs, they are having a huge size that is more than 200 nanometers or something, then what happens is that they will be taken up by your antigen presenting cells and they will be cleared off. There is no use of such a mRNA LNP vaccination. But if your size of particles is too small, let's say less than 50 nanometers or something, then what happens is that they can be easily cleared off by the kidney, and that is the reason that we keep the mRNA LNP size around 100 nanometers. That is the best size that you can keep for an efficient delivery and immune response. Talking about the sterols, this is the outer component of the LNP, and the modification changes the LNP organization at the surface. So replacing the cholesterol with naturally occurring phytosterols such as beta cytosterol and oxidized cholesterol derivatives, it can improve mRNA delivery. Okay, so because their polymorphic shape and multi-lamellar organization can deform the endosomal membrane or extend the time frame of nucleic acid release. Then phospholipids, like you can have the saturated highly melting point phospholipids such as DSPC. It can result in the production of highly stable LNP but will not facilitate endosomal escape. Then you can use unsaturated low melting point phosphatidylcholines such as DOPC that may yield fluid LNPs that are highly susceptible to opsonization by the serum proteins. So they are usually combined with the cholesterol to formulate highly stable LNPs, okay, that can maintain their integrity. So here you can see a graph of the size of the LNP as well as the molecular weight of the pegylated lipid. So that is something that you can see that as you are increasing this mole percentage of the peg lipid, then the mean particle size or the diameter that is there of the lipid nanoparticle, that is decreasing and it is becoming constant at a particular threshold. Then if you talk about the formulation, now you can ask me that how we can do the formulation, how we can like combine the lipid, LNP and everything. So there are different methods for formulation. The first one is a thin film hydration method. Okay, another one can be a ethanol dilution method, which is most widely used. So talking about the thin film, you can have a lipid and other excipient that is dissolved in the organic solvent. Then you evaporate the solvent, and because the lipid is dissolved in any organic solvent, it can, when once it evaporates, you get a thin lipid film in the flask. And then you can add the aqueous solution containing your drug or mRNA, that is mainly your citrate buffer containing your mRNA. Then the liposomal formulation containing the MLV, it will be there. And because there is a lipid film, when you add this, these get encapsulated, and then you can use sonication or something for reducing the size, because the film was all over the flask. So the size of the particles formed over here will be very large, very huge. So you have to reduce it. So for that, you can use high frequency sound waves to reduce the size of the particles. So you will have the liposomal suspension. So this is one method. Another method is a very widely used ethanol dilution method. In this, you are having a lipid mix. Okay, so you are having the solution on organic solvent containing your ionizable lipid, your helper lipid, cholesterol, PEG, ethanol. Okay, mainly you are using ethanol as an organic solvent to dissolve the lipids, and your mRNA is present in the citrate buffer. Okay, now you have some microfluidic devices. Okay, so these microfluidic devices, these are narrow channels which are present on a PDMS or a kind of polymer, in which there are two paths. You can see one is this and one is this path. So when your lipid mix of the ethanol, it enters into this path, and your nucleic acid or mRNA in citrate buffer, it enters through this path. What happens is in between you are having a kind of a mixer system, the microfluidic mixing system, which can give a different, which can flow these in different pressure rates at a different ratio, and at a particular pressure rate there is a formation of the lipid nanoparticles, because these two combine, they coalesce together and they form the LNPs. Okay, and then your LNPs are present in the citrate buffer. You can call like have buffer exchange in PBS of pH 7 to keep them neutral, and then you can check for the encapsulation efficiency. So how you can check for the encapsulation efficiency? You can run an agarose gel in which you in the two wells you are having one as the unlicensed formulation, in which you are just directly loading the LNP mixture, and in the another well you are having a equal volume of the lysed formulation, in which you can add some detergent, that is Triton X100 or something, and you can load both the gels along with a ladder in 1.2% agarose gel, and you can check that with respect to the unlysed what amount of uh, mRNA you can see in the lysed fraction, and then you can subtract their relative intensities to get to know about the amount of mRNA that is encapsulated inside the LNP, because the one which is lysed, it has all the mRNA content that is present inside the LNP as well as some that is unformulated that is present outside. But in the unlysed fraction, you are having only the content of mRNA that is not encapsulated, that is present in the solution outside the LNP. So now once you are subtracting that background noise, the unlysed one from the lysed one, you are getting the amount of encapsulated mRNA. Okay, and you can calculate the encapsulation efficiency by the similar way that you subtract the lysed minus unlysed, divide by the lysed amount of mRNA, and you multiply by 100, you get the what you call as the encapsulation efficiency. Then you can check the size and the zeta potential of the mRNA. How you can do that? So to check the size, you can use a technique called as dynamic light scattering, that is DLS, in which you can get to know about the size as well as the zeta potential as well as the PDI value. So what is PDI? PDI is polydispersity index, that represents that how homogeneous your solution is, that how much, like, you can say how much variation is there in the entire formulation. That let's say you are in the PDI, so the accepted PDI values are zero point 0.2, below zero point 0.2. Okay, so if you're getting values below zero point 0.2, that means that your formulation, it consists of LNP which has encapsulated mRNA of a particular size range of 100 nanometers only. But if you are having a lot of LNP aggregation and you are having some LNPs of size 200 nanometers, some LNPs of size of around 500 nanometers, and some LNPs of sizes of around 80 to 100 nanometers, then you will get multiple or broader peaks in your PDI. So that indicates that your formulation is highly heterogeneous and that is not acceptable for any immunization study or to carry it forward in the clinics. Okay, so that is something which is important. And then you can have some other methods also, like rapid mixing, in which you just keep your aqueous mRNA solution in citrate buffer and lipid solution in ethanol, and using pipettement you just pipette them rapidly, and that pipetting can also lead to formation of the LNPs. Okay, so that is also something that you can do. Now we talk about the desired properties of the mRNA LNP. So I have told you that size of the LNP usually measured by the dynamic light scattering. It should be around 20 to 200 nanometers, but mostly we prefer it to be around 100 nanometers. Okay, and so as the size makes them robust to withstand the fluid flow, that is example blood and lymph, while allowing LNPs to cross the interstitium, then LNP surface charge, it should be neutral to avoid any unwanted electrostatic interaction, but should acquire a positive charge at acidic endosomal pH. So if LNPs have negative charge, they won't be taken up by the negatively charged cell membranes. And that is why if it has positively charged, it will disrupt the cellular membranes and cause cytotoxicity. So, but commonly in the zeta potential we have measured is that the surface charge is considered weak if the zeta potential falls below between minus 20 to plus 20 millivolts. Okay, and the N to P ratio. Okay, what is N to P ratio? It represents the nitrogen to the phosphorus ratio, that is of the LNP. Okay, of the head groups and all your mRNA. So this N to P ratio, that this adjusts the total surface charge of the LNP. Okay, and then you can have these. So if you talk about the lipid packing, so based on the N to P ratio, if it is 2, 3, 4, 5, or 6, there can be variation in the LNP diameter, and it can range from around 200 to 100 to 50 nanometers. And if you talk about the encapsulation efficiency, so with the increase in the N to P ratio, you can see a rise in the encapsulation efficiency as well. Okay. Now, apart from all these things that we have discussed, one thing is important, that is preservation and storage conditions. Okay, so two main vulnerabilities of the formulation is the aggregation or the leakage or the fusion issues of the LNP. So what is important is to degrade. What is important is to be taking care of the incorporation of the stabilizing lipids and PEG, and adding some cryoprotectants like sucrose or trehalose, which can lead to protection of the overall formulation. Also, there can be degradation of mRNA by the co-encapsulated water. So a tighter encapsulation, that means encapsulation that will only act exclusively towards the water molecules. Okay, and mainly it will keep the payload as dry. That means you don't have any water inside the LNP. You are only having your mRNA and slight water to maintain it. So that is something that is desirable. And you can see over here that is the luciferase expression for the different kinds of cryoprotectants that have been used. So these are the fresh LNP. They have very lesser expression, but you are having this kind of 0% sugar, 20% trehalose, 20% sucrose, then the expression you can see over here. So recently there have been studies where they have used some kinds of other cryoprotectants which are better than the sucrose, sugar, or trehalose, like betaine. Okay, so you can read some papers on it that the incorporation of the betaine. So there was a paper in Nature Communication where they have said that when you incorporate betaine into the formulation, it is better than all the sucrose, trehalose, and sugar as well. So that is the latest thing that you can read about. Okay, so for long-term storage, freezing, drying, or lyophilization, you can use betaine. Then we have talked about the modifications. So the incorporation of the chemically modified nucleosides like pseudouridine, thiouridine, or 5-methylcytidine, okay, that is being done in Pfizer and Moderna formulation. It can significantly reduce the innate immune response and increase the adaptive immunity by the mRNA translation. If you optimize the sequence by incorporating frequent codons, like the codons with a higher tRNA abundance, you can increase the protein expression, you can enrich the GC content, and you can increase the steady state mRNA level, okay, which can act as a self-adjuvant and activate the innate immune response and giving you a better immunogenicity. So in terms of nucleotide modification, there is a trade-off to consider between potential innate adjuvant responses driven by unmodified nucleosides as well as the enhanced protein expression that is attributed to the modified nucleosides. So, in phase 2B or 3 clinical trials, the CureVac's efficacy was found to be less than Pfizer and Moderna due to different conditions, including unmodified nucleosides, different UTRs, not any significant optimizations that are being done, and storage conditions. Now there is an important concept of RNA capping. That is something which should be discussed. So if we talk about the RNA capping, what you have is that you all know that there is a cap which is present on the mRNA that gives it stability. Okay, the 5' cap which I have talked about earlier also. So if you see these caps, what happens is that you are having something called as a cap 0, cap 1, and cap 2 structure. Okay, so mainly what happens is that eukaryotic mRNAs, they have some 7-methyl guanosine cap, okay, that is linked to the first nucleotide of the mRNA by a 5' 5' triphosphate bridge. Okay, now chemically you can write it as something that is, you can call it as M7 GPPPN. Okay, so the normal mRNA, it consists of your 3' to 5' phosphodiester bonds, and the cap has a 5' 5' linkage. Okay, so if you talk about, so because of this, it cannot be recognized by exonucleases and it protects mRNA from degradation. Now if we talk about the cap 0 structure that we also called as a minimal cap, what happens is that it has this, so basically here N represents A, G, C, or U. Okay. And in case of cap 0, you are having only one methyl group which is present, which you can see over here, that is added at the N7th position of the guanine, and there is no ribose methylation on the first nucleotide. So what happens is that when the enzymatic removal of the gamma phosphate happens from the 5' end, the addition of GMP happens via the 5' 5' triphosphate, and then there is a N7 methylation of the guanine. Okay, so this is recognized by the translation machinery, and still it is sensed as non-self by the innate immune receptors. Okay, so cap 0, it is common in yeast, in some viruses, or some in vitro transcribed mRNA. Then comes the structure of cap 1. So what is special in the cap 1? So the cap one, if you want to represent this, you can write it as, so if I write cap one can be written as your M7 GPPPNM, okay, where NM refers to the first nucleotide. Okay, this refers to the first nucleotide with a 2' O methylation on ribose. Okay, with a 2' O methylation that you can see in this figure, the 2' O methylation that is present in the ribose. So that is something we represent as the cap1 structure. Okay, so there is an additional methyl group at the 2' hydroxyl, and this does not change the base pairing, but it changes the steric and electrostatic properties. This 2' OH 2' O methyl group, it reduces the hydrogen bonding potential, increases the hydrophobicity at the 5' end, and it also alters the RNA backbone geometry. Okay, so you can prevent the recognition by the RIG1 and other kinds of innate immune responses which are there. So cap1 is the dominant cap in mammalian mRNA. If you talk about the cap2 structure, here you are having a methylation at the second nucleotide ribose. That is, if you want to represent the cap2 structure, you will simply write this M7GPPPNMNM. Okay, that means at the first and second nucleotides, they have the 2' O methylation. Okay, so that is something you can represent these structures. And the key chemical difference is that there are two ribose methylations now. Okay, it further reduces the backbone flexibility and it causes a stronger steric hindrance at the 5' end. Okay, so it further suppresses the innate immune response and stabilizes the mRNA protein interactions. Okay, and it is seen in highly expressed or long-lived mRNAs also. So this is something that is there. There is a difference in the methylation pattern and the number of methyl groups which are present. Okay, so why these chemical differences matter? Because the rig1, it detects the 5' triphosphate, okay, and the unmethylated ribose, and the 2' O methylation, it disrupts this detection. Okay, so that is something that is there. And if you talk about the effects of capping, so 5' capping, it helps in binding to the elongation factor and to improve the translation, and it protects from 5' to 3' exonuclease cleavage. So reverse capping at the 5' end, it induces the rapid degradation of the mRNA. So the capping, it can be co-transcriptionally capped using cap analog, and it can also happen post-transcriptionally by enzymes. Okay, so multi-step enzymatic capping, it requires more product handling and incubation at higher temperatures to de-ensure the secondary structures at the 5' end for enzyme access and to degrade the mRNA. Okay, so if you're having a co-transcription capping strategy, so then your DNA template should have RNA polymerase promoter sequence, initiator sequence for cap analog, 5' UTR, ORF, 3' UTR, and a poly DT tail for transcribing poly A. Okay, and in RNA's free condition, the linearized template. So basically, how we do IVT, that is in vitro transcription, we have a linearized template which can either be by single digest of your plasmid DNA or by, you can say, PCR amplification of that particular region which encodes for all these regions. Okay, once you have the linearized template, you add the necessary NTPs, which can be ATP, GTP, CTP, and N1 methyl pseudouridine as a modified base analog for uridine, and you can add the cap, which can be this clean cap that can be used, and the transcription buffer. Okay, and the RNA polymerase, that is the T7 RNA polymerase, they are added to one pot to generate the capped mRNA of interest, and then DNA can be added to the final IVT mixture to avoid any kind of DNA contamination. That is the linearized template we are referring to. And the mRNA you can purify, you can use sodium acetate purification or lithium acetate purification, and you can store it at -20 for short term and at -80 for long term storage and uses. So these are the structure of the different caps. That is the ARCA cap, anti-reverse cap analog. That is the cap 0 structure with G as a +1 nucleotide that I wrote over here. You can see. Okay, that is the N part. Okay, and you can also have this clean cap which I was talking about, that mainly consists of your cap 1 structure with A and G. Okay, with A and G as your +1 and +2 nucleotides respectively. Okay, so you have these advantages of clean cap over the ARCA cap, that is hybridization of the +1 nucleotide of ARCA dimer, that is G with template, is crucial. There is a competition with the GTP and there is a reduced yield. Okay, and the truncated mRNA can be due to the limited GTP and there can be inefficient capping that can happen. Okay, so there is a need for purification of immunogenic 5' phosphate mRNA, but the clean cap AG trimer, it has two nucleotides to bind to the template. That is, there is a lesser competition from the NTPs and it is entropically more favored. So there is a more efficient capping. There are lesser chances of truncated mRNA products. That's why clean cap AG is better over the ARCA cap to be used in the IBT. Then the cap1, this is another paper which represents that the cap1 messenger RNA synthesis with co-transcription clean cap analog by in vitro transcription, where there is a reaction yield that is mentioned, and mainly you are having this gel. Here you can see that the lower molecular weight smears are the RNA degradation, and higher molecular weight smears, they represents the presence of the salts. So what happens is, you can see over here that with the help of this clean cap, you are getting a better and efficient yield of the mRNA. Okay, and there can be other modifications of the mRNA, like you can modify the 5' UTR. Okay, you can use the UTR of a tobacco virus or a human heat shock protein to increase your translation efficiency. Again, I told you there are certain AI models now like RiboNN and other models which are predicting the better UTR sequences as well as the ORS sequences. Then in the 3' UTR, you can have the beta globin 3 UTR, because globin's mRNA produce large amount of protein and have longer half-life. In case of poly A tail, you can reduce the RNA exonuclease activity, you can stabilize the RNA, you can bind to the PABP triggers, microRNA, mediated inhibition of translation, and the length of the poly A tail needs to be optimized to get your desired protein expression length and can be specified by incorporating specific length of the DT region in the DNA template instead of non-specific enzyme modification. Now, again, we have told you that about as the LNP, it can be used as a delivery vehicle. So, due to the dynamic stability, biodegradability, and endosomal escape, and these were the different components of the LNP. This is the overall structure of the LNP with the pegylated lipid, charged ionizable lipid, cholesterol, neutral ionizable lipid, as well as the DSPC. So, the ionizable lipids basically, I told you they have a head group linker and the lipid tails. This is the ALC0315 from Pfizer or BioNTech. This is from SM102 from the Moderna's mRNA-1273, and there are different lipids. So about the head group, I have told you what are the parameters which you have to look into while designing the head group, the linker and the lipid tails. Okay, so um, this is something that is important. And if we talk about the other constituents, that is a PEG, it increases the systemic circulation. Okay, and then your helper lipids, mostly your sterols and phospholipids, they are called as the helper lipids. Phospholipids being cylindrical, they form the bilayer structure, increases the TM of the LNP, that is the fluid LNPs are more susceptible to the serum proteins, and cholesterol can fill in the gap between the phospholipid and stabilize the bilayer. Both cholesterol and phospholipids, they are natural components of plasma membrane and promote membrane fusion and reduce the toxicity. So if we talk about the long-term storage, so we have to like prevent the mRNA from getting degraded. We can use cryoprotectants, that is what I told. And now to conclude my talk, basically we have seen that mRNA-RNP vaccine is an effective modality due to its rapid and highly scalable manufacturing process, and there is a to maximize the protein expression, both mRNA sequence and lipid LNP composition needs to be optimized. There is a weak correlation between protein expression level and immunogenicity. So in terms of efficacy of mRNA vaccines, the ability to elicit immunogenicity, it should be the endpoint of screening the formulations. So assuming only two dose vaccines are prevalent over the market, it would need 11 billion vaccines to vaccinate 70% of the world population. Okay, so this is the fact that is really astonishing. And so the requirement for storing such huge amount of vaccine at ultra low temperature is challenging. So that is why currently novel solutions for mRNA LNP stability at non-freezing temperature is still in demand, and possible solutions are use of cryoprotectants. So recently you can check out that paper in Nature Communications on using betaine in the mRNA LNP formulation for this purpose. And basically lyophilization and nucleotide modification can improve the stability. So, although mRNA is a promising therapeutic molecule, its full potential can be utilized if it can be protected well from the physiological barriers, as well as it can be released at its target site. So, the LNP formulations, mostly due to its non-toxicity and high encapsulation efficiency, has come out to be the most effective choice of vectors. Okay, and the stability of the entire formulation can be modulated by varying the lipid compositions, N to P ratio, etc. But there is not much concrete studies on mRNA LNP formulations. Okay, so there is a knowledge gap, and I hope that some of us will fulfill that gap. Okay, so yeah, so this was about my talk, and if you have any questions, you can ask me.
Hello, sir. I have a few questions, if you don't mind.
Yeah, yeah, please ask.
So, how do you optimize the mRNA and the UTRs and the and the mRNA? Like you mentioned about it optimizing, right? How do you do that?
Okay, so for the optimization purpose, what you can do is you can just search in the Google about a paper on riboNN published in Nature Nanotech. Okay, so there are different AI models and there are different neural networks. What they do is they are trained on the data set of highly optimized mRNA sequences. Okay, so if you are having the data of all those sequences, let's say in your liver is producing proteins, they are highly expressed proteins. So what is special about the mRNA of those proteins in your liver? What are those patterns? What are those codon usages? So it is trained on all those data sets, and then if you generate, you can like give it a particular sequence and it will give you an optimized mRNA sequence. So it generates an optimized sequence based on the weights that are given to different parameters. These parameters mostly they can be like the occurrence of different codons, the codon usage, as well as the patterns of codons which are found in highly transcribed genes. So based on those patterns, it can give you a better optimized sequence. So mostly the optimization is done with the help of these AI models in current times. Before the AI models and all these neural networks, people were simply making library of mRNAs. They are mutating the sequences and they are checking which sequence is giving a better expression and all those things. So you can do today in a very quick way by using these models.
Okay, so now optimization is turned into dry lab basically.
Yeah, basically you use dry lab, you generate a lot of sequences, then you check them in vitro whether or which one of these predicted sequences they are yielding you better expression of your protein on the cell surface.
Okay, sir, I had one more question. Does
Yeah.
these mRNA vaccines have any restrictions with the type of disease?
Okay, restrictions as in like.
Let's say, does it work for any type of disease or any type of disease?
So the thing is that the application of the mRNA vaccines they have been also extrapolated to cancers and all this because the majority of the purpose over here is that you simply your body will synthesize proteins. It has the machinery. You just provide it with the particular mRNA that you want to display on the surface of the cell. So now how you display on the surface, you use some transmembrane domain that is present naturally in these viruses. And once the transmembrane domain is linked to the particular sequence of your viral protein, what will happen is that on the cell membrane, your protein will be displayed. And once your protein of interest is displayed on the cell membrane, then you can simply have any particular like encounter by the other immune T cells, and it can lead to generation of your cell mediated immunity. So this is the concept. Now the challenges occur on the folding of that protein on the cell surface. There can be restrictions because like some proteins they don't fold well. Okay, and once you fuse it with the transmembrane domain, then there can be a different conformation that the epitopes which are recognized by the T cells, they are not accessible. Okay, so these are some restrictions which are there in case of different viruses, but mostly in case of SARS-CoV-2, it has proved to be a better vaccine candidate.
Okay, sir. Got it. Thank you.
Welcome.
In the essence of time, we will only take one more question now. So anyone, you can like unmute yourself, or you can write it in the chat.
Hello, sir.
Hello.
Hello, sir. First of all, thanks for the wonderful session. I just have one question that is during molecular RNA vaccine development, how do team decides when to pivot versus persist with a platform that shows mixed results? Because the early stages data might be confusing.
So basically, what we do is we do a lot of studies before moving it further. So the primarily study starts with in vitro expression. So what we do is once you have made the mRNA, first you check the mRNA, you just transfect it, you do a transient transfection into some cell lines like HEK293T, and when you transfect the mRNA, you transfect with the help of some reagents like lipofectamine, which make a lipoplex kind of thing outside the mRNA. It is delivered into the cell, it is expressed on the cell surface. Let's say you are expressing the RBD region of the SARS-CoV-2, then you are having some antibodies which bind to the RBD region. So once it is expressed, you use flow cytometry assay, you check whether your protein is on the cell surface or not. Then you can use the antibodies, okay, specific to the RBD like S309, and then you can use a secondary antibody like anti-human Elexaflor 633, and you can detect the presence of your protein. Once that is done, then you encapsulate inside the LNP. Simply you deliver that LNP into the cells, and then you check the expression. Once you are getting expression, now you can carry out a what do you call as a immunization study in the mice. You immunize the LNPs, okay, mostly intramuscularly, and once you immunize, you check the after the prime and the boost immunization whether there is a elicitation of the immune response, whether there are neutralizing antibodies or not, okay, and then you can also isolate the spleen, and then you can check for the in the splenocytes you can check for the different kinds of markers of the immune response like the IL-6, IL-3, TNF alpha, all those markers, whether it has elicited a T cell immunity or not. And once you're done with all these checks, you can also check the localization of the LNP, that whether where it is going. Okay, it is concentrated in the spleen or the liver, or it is going somewhere else also. You can check all that. And once you're done with all these checks, then you can simply rely on your approach.
Thank you, sir.
Welcome.
Okay, so with this, we are going to close the Q&A session round, and now like closing the session with the vote of thanks. So thanks, Mr. Deepesh Joshi, for such
Thank you,
an
sir.
interactive and insightful session.